Okay, good afternoon. Next, I'm going to give an update on the ICH M13A, which is the first ICH guideline for bioequivalence. And you may be aware from yesterday's opening remarks from Commissioner or from the news, this really represents the milestone for the generic drugs in terms of being in the global level. So I will provide an overview of the M13 Guideline Series, not just M13A, but what's the plan, and also provide an overview of the final M13A guidance and major changes from the draft, and also what the major difference from the FDA current general guidance, as well as provide an update on the FDA planning on the implementation of M13A for the generic drug development. So for this audience, you all are aware the bioequivalence assessment is really very important for the establishment of the therapeutic equivalence for generic drugs to their respective reference list drug. And also you may be aware the reference list drug is the terminology FDA is using, but for the M13A, comparator product was utilized. It's equivalent to FDA's RLD, which represent the standards, this is the terminology we harmonize at the global level to represent the reference standard other regulated agencies will recognize for their regulation needs. And we also know BE is not only for generic drugs, they also be utilized by new drugs for their decision-making, either pre-approval or sometimes the post-marketing, post-approval formulation or manufacture changes. So it's very important a concept. However, traditionally we know in the ICH level, there's no harmonized guidance for bioequivalence or for generic drugs. So this is really the first topic bring to the ICH. Starting with the reflection paper that was adopted by ICH in 2018 on further opportunities for harmonization of standards for generic drugs, which laid out a strategic approach on how we harmonize and provide these scientific technical standards for BE, starting from a more simpler product to more complex products. And the ICH also formed the generic drug discussion group following the reflection paper. The discussion group really have representations from not just the regulatory agency and the ICH, but also the industry groups to recommend topics for harmonization and what should be the order for prioritization. And they also supported the submission of the first new topic proposal, which is lead to the M13 expert work group formation, focus on the immediate release oral solid dosage form. And the M13 expert working group was formed since 2020 and mainly focused on immediate release solid oral dosage form. And based on the scope of the work, the EWG proposed to develop M13 in 3 series. So M13A really represents the first guideline under this series, focusing on the BE study design and analysis for immediate release solid oral dosage form. And later there will be a M13B, is second in the series, will cover the topics on additional strength as well as including the additional strength waiver. And the M13C will cover the data analysis and BE for highly variable drugs, therapeutic drugs, as well as some more complex study design and data analysis, including adaptive design. The reason to break this into 3 series, so the working group will be able to break down the topics so we can deliver those finalized guidance in a timely manner, in sequential, but also consider there could be some overlapping while we work on those guidances in order to bring the efficiency. So right now the M13A is finalized. We are very happy. Now it's reached the step 5 and it's ready for the global regular agency to implement this guidance in the generic drug development. M13B is ongoing, and the working groups currently work on reaching consensus on the draft technical guidance to anticipate to reach step 1 towards the end of this year. And the M13C has not started yet. It will start after M13B is the draft is published and during the public consultation stage. So it is really a long journey when I'm looking back. I have the privilege to serve as the rapporteur for the M13 expert working group. And Lufa Temple, who will be on the panel, she is the FDA topic lead. We have a very diligent and collaborative global working group under ICH with 22 members, observers, as well as among them, 14 of them are the regulatory agencies. So we have been working very hard in the past 4 years by published this guideline follow the ICH process for harmonization. Really, there are 5 steps as laid out in this slide. And we delivered the draft guidance toward the end of 2022, and we received a lot of public comments, and we reviewed all of those comments and finalized guidelines based on the comments received and understand the areas that may need additional clarification. And the final guidance was finished and adopted by the ICH in July of this year. And now it's ready for, at step 5, ready for implementation. And the global regulatory agency under ICH also have their own plan for the implementation, and the FDA is also in the process of revise our general guidance as well as the product-specific guidance to align with M13A. So the overall content of M13A in the final guidance does not change from the draft guideline. There's 5 sessions, including the introduction that will cover the objectives, background, and scope. And the key content is in the section 2, which talks about general principles for establishing BE with both study design and the data analysis part. And we focus on non-replicated study design. And we also have 4 sections covers various specific topics on endogenous compounds, other immediate release dosage forms, which including the oral disintegrate tablets, chewable tablets, and oral suspension, as well as the fixed dose combination. And we also have the session talk about the pH dependency, when you may need to consider one additional study under the with a acid-reducing agent in order to establish BE. And we also have a section on documentation and glossary. So during the public comment set period, we received close to 800 non-replicate comments, and the majority of them are related to Section 2. It's not surprising because this is the key part of the guidance. We have 77% comments related to that section. In addition, we have 10% of comments also related to Section 3. And the major comment themes are focusing on, because we introduced the risk-based approach for determining whether we need 1B studies or 2B studies under what fasting or fed conditions. So there's a lot of clarification needed on how you define high risk and when fed B study is needed. We also received some other comments related to how you define the dose proportionality. This is also related to M13B in terms of additional strength waiver, and also study population, data analysis, various aspects of it, group effect, and also alignment of study design with product labeling. So the expert working group reviewed all the comments, and the modifications are being made to based on those comments received. And also, in addition, we also realized some topics may need additional clarification or rationale, which we don't have the space in the guidance to provide that. So when we finalized the guidance, the working group also developed a questions and answer document. It's published the same time as the final guidance. So we don't think there's a major changes from the in terms of the recommendations from the draft guidance, but we did make modifications on several sections, mainly to provide clarification to help the implementation or make them more practical for the regular agency to implement the guidance. So in particular, the high-risk product, what does the risk mean in this under the context of M13A? It is really to mean the risk of the B inequivalence or B failure for those products which has special characteristics. They may result in a high risk of the B inequivalence under the fed condition due to the food effect differentiation differential food effect caused by formulation or manufacturing process. And also the data analysis part has some more clarification provided, as well as PK dose proportionality assessment, early exposure. We, in addition to the partial UC, we also introduced the Tmax may be used, and also additional clarity on pH dependency when you may need an additional study, and what justification you may provide. So as I mentioned, the working group provide additional questions and answers document to help provide further clarity and also assist the implementation. As you can see, all the Q&A are focusing on section 2, 3, and 4. In addition, as part of the ICH process, a step 4 presentation was published at the same time. This is serving as the basis for training materials to help the implementation, and the links on the slide. So if you read the guidance, before the M13A, the different agencies do have, even the B has been used for many years. The regular agency globally do have different recommendations for B study design and the criteria to support the generic drug approval. This may result in inefficient drug development, increased cost and time. Additionally, maybe cause some drugs may not be approved in certain market. And the major difference we are aware is the FDA generally recommended both fasting and fed studies. However, several other regular agencies, including the European Medicines Agency, EMA, they generally recommend one study, which is fasting studies. So those differences in the general B guidance can lead to the different product-specific guidance recommendations, which both FDA and EMA has PSGs developed in addition to the general guidance. So this table just really to show you those differences in the general guidance does translate to the PSG recommendation. If you look at the comparison between US FDA and the EMA, in terms of PSG, although FDA do develop more PSGs than the EMA, but if you look at the percentage-wise, we do have a majority, like 86% PSG prior to M13A recommend both fast and fast studies, while EMA only have 16%. And they have majority recommend the fasting studies only, because this is reflected by the general B guidance differences. So as we are aware, the M13A introduced the risk-based approach to determine the need for the B study condition. So for high-risk product, as I briefly alluded to, those are the products which difference in the formulation or manufacture process may not be detected with a single B study, either in the fasting or fat study. Fat condition may not be extrapolated to predict B under other conditions. Therefore, the guidance recommend the B study for those type of product. B should be conducted in both fast and fed conditions, and also Q&A touched further rationale on why we recommend this. And for non-high-risk products, on the other hand, the results from a single study, which typically could be fasting study, could be extrapolated to predict fed B study outcome or vice versa. So the B test only one condition is sufficient. And the guidance, the revision, you can see there's some slightly different language used in the final guidance, but the concept is not really changed, but we just try to articulate better how you identify a product as a high risk, non-high risk, what you may consider when you develop your product to consider whether you need to do 2 studies versus 1 studies. So really we need to look at the reference product or ILDs, drug substance characteristics, coupled with their formulation, design, or manufacture process, and what's the labeling used, what's the food effect, how they those with relation to food, and there's specific product formulation that consider high risk, which a couple of examples were given in the guidance, which include the solid dispersions, microemulsions, lipid-based formulation, nanotechnology, and some other specialized technology. And also in the guidance also alluded that the test product also need to consider how you similar to the reference product, just because some product or some reference product may not deemed to be high risk. However, although some product may not use the complex formulation, but the specific characteristics in the formulation, if it's different between the test and the reference product, they can result in a formulation that may modulate the food effect different, and there might be a need to conduct a study under both conditions or provide justification. And also in the guidance, both in the draft and also in the final guidance, talk about the meal type for Fed B studies. So for high-risk product, the guidance recommend in respective of the product labeling, the study need to conduct both fasting fat, and the fat, it will be the high-fat, high-calorie meal. However, for the non-high-risk product, if the Fed B study is needed, especially when that's where the labeling indicates the drug should be taken with the food for PK reason, then either a high-fat, high-calorie meal or low-fat, low-calorie meal can be used, administered, and also labeling. And also if the labeling specifies certain meal, that could be the meal used for the B studies. And also the additional rationale we'll provide in the Q&A. So this kind of slide summarize the current FDA B guidance versus the M13A in terms of fasting and fat B studies. So FDA gonna change from the current recommendation, which is for pretty much for almost all oral IR drug products, asking both fast and fat B studies, will move towards the risk-based approach as delineated in the M13A. And as I already mentioned, so for the high-risk product, probably both fast and fat study, but for non-high-risk product, they could be either fasting-only study or fat-only study, or either fasting or fat. Depends on the situation as delineated in the guidance. So FDA is really leading the harmonization efforts, and ICH recognize the importance of the harmonization in bringing the access to the medicine globally. And because of the M13A now being finalized and adopted, and FDA will revise majority of the IR oral products, PSGs, to align with M13A, following the risk-based approach, by considering the BCS classification of the drug substances, formulation design of the ROD, and what's the food effect, what's the labeling condition, whether the drug is to be taken with food or without regard to food. So based on this assessment, we look at more than 1,100 PSGs, current existing PSG, which covers those products. And we recognize that more than 800 of these PSGs can be determined as non-high risk. So we will recommend from the current 2B studies to 1B studies, either in the fasting conditions or fat conditions, which means we will remove either fat B study or fast B studies based on the labeling condition. And you can see we published the forecast list. There's a link down there last Friday, so you will see what are those 800 plus PSGs are. And we also aim to publish the revised PSG in the first quarter of 2024, which target in October next month. So the significance of M13A is very obvious. So this is the first harmonized ICH guideline focused on BE. It will impact a significant portion of the pharmaceutical market. This is also the reason why the ICH start the IR product first, because it's almost occupy about 50% of the market or approved new drugs in the US. And it will reduce the need for additional in vivo B studies due to the divergent recommendations. And it will support a more streamlined global drug development. Although I did not really cover much, is the M13 also recommend a study design where multiple arms can be utilized in the study design to include multiple reference products or comparative product or multiple test product in order to improve the efficiency and satisfy the multiple jurisdiction requirement. And this will benefit the patients by increasing access to generic drugs. So here are the key takeaways. So M13A has been finalized, and it's adopted by ICH, and it will be implemented or is in the middle of implementation for ICH regulatory members, including FDA and other regulatory agencies. It will facilitate the regulatory approval, promote confidence in the quality of the generic drugs, and also enhance global access to those medicines. FDA is updating the relevant guidance, not just the PSG, but our general guidance will also be updated to align with M13A, with relate to the IR part. And also just remember, both FDA's current guidance and ICH M13A do offer flexibility. The ANDA applicants may provide appropriate scientific justification if they decide to take an alternative B approach, different from the guidance, deviate from guidance. That is also possible. So today I only can have a very high-level update on this important guidance, but we plan to have a SBIA webinar on this guidance on November 21st, save the date, for a 2-hour webinar, which will have additional detailed update on the key scientific thinking behind the recommendations, FDA implementation plan, and address your questions more thoroughly. Now is the challenge question. Challenge question number 1. Following the implementation of ICH M13A guidance, which of the following will represent the most significant changes in the FDA recommendations for B studies for immediate release solid oral dosage drug products? A, B studies designed with regard to food conditions, B studies population, C, B for long half-life drugs, D, batch size for pivotal B studies. So the correct answer is A, B study design with regard to food conditions. Challenge question number 2. FDA will revise product-specific guidances for immediate release oral drug products to align with ICH M13A. Is true or false? I hope you get it is true. Thanks. So here are the resources. Hope we'll provide you additional information. And I would like to end that we, as OGD, as FDA, we collaborate globally to resolve scientific and regulatory challenges to ensure access to generic drugs. With that, I'm going to turn over to Yu Qin, who will provide an update on the MIE program, pilot program. Thanks.